Welcome to the third Systems Biology practice session. A valuable resource is DrugBank. DrugBank is an online database containing information on drugs and drug targets. How can we find drugs on DrugBank? We would like to find the drug that targets the IDH1 protein we've seen in the previous practice sessions. If we type the drug name and search, we have only one result, the drug Ivosidenib. Let's look at the drug information page. The page is divided into sections, and you can navigate using the toolbar on the right. You press the arrows here, and you can jump to whatever section you'd like. The first section is identification, and it contains general information about the drug. We can see that the drug is approved for therapy. In the description section, we have information about the drug. We can see that it was approved by the FDA and targets IDH1. We can also see that it has a coupled companion diagnostics, and we will elaborate on this later. This page has a lot of information. We can see the structure of the drug. In synonyms, we can see if the drug has other names. On prescription products, we can see the commercial name of the drug, and that in this case the drug is an orally given tablet. In categories, we can see families that the drug is part of. For example, it is a cytochrome P450 CYP2B6 inducer. We can also see different identifier of the drug and its molecular weight. In the pharmacology section, you can find information about the conditions treated with this drug. In this case, it's refractory relapsed acute myeloid leukemia. We can see the mechanisms of action of this drug, data about absorption, cleaning, and toxicity. In the interaction section, we can see other drugs that can interact with our drug and the effects of this combination. The references section shows a list of relevant literature, links to other databases that has information on the drug. Since this drug is FDA approved, there is a link to FDA label and the material safety data sheet link. Next, we can see information regarding clinical trials, their phase, status, purpose, and conditions. If available, the pharmacokinetics section shows the price, manufacturers, dosage forms, and registered patents for these drugs. After the last section, taxonomy, which in this case is not available for this drug, we can see the information about the target. In this case, isocitrate dehydrogenase, NADP cytoplasmic, and the gene name, as we already know, is IDH1. To conclude the first part, DrugBank contains very detailed information about drugs, their properties, mechanisms of action, and other molecules they interact with. Now, I want to go back to something we skipped in the beginning of our session. You may remember that in the description of the drug, there was a passage regarding companion diagnostics. It says, this approval came alongside the approval for the real-time IDH1 assay, which is meant as a companion diagnostics tool to detect IDH1 mutations. What does this mean? FDA companion diagnostics are tests that analyze variation in the sequence, structure, or expression of DNA and RNA in order to diagnose disease or medical conditions, infections with an identifiable pathogen, or determine genetic carrier status. These tests help us to determine whether a drug should be administered to a patient or should not, based on specific genetic information. Let's enter the web page for nucleic acid-based tests, as we wish to find companion diagnostics for the IDH1 gene. So this is the website. The easiest way to find the companion diagnostics for the gene and drug we saw on DrugBank is to search the page with Ctrl F and type IDH1. We have only one result, for the treatment of acute myeloid leukemia. Let's press on the submission link. We can see that it is a somatic gene mutation detection system. In the approval order statement, we can see that it is an assay for the qualitative detection of single nucleotide variants, SNVs, coding 5 IDH1R132 mutations in DNA extracted from the human blood or bone marrow. That means that instead of the regular arginine that is supposed to be on the 132 position on the protein, there are 5 different possible mutations: cysteine, histidine, glutamine, serine, or leucine, and patients with one of these mutations are recommended for the treatment with ivosidenib. The companion diagnostics identify patients that can benefit from the drug and prevent us from administering drugs to patients that are not likely to benefit from them. It is a very useful tool in planning the course of a treatment for a patient. What happens if the gene we wish to target does not have companion diagnostics or any known drugs? In that case, we can also search for drugs that target other genes from the same pathway using KEGG, or find associated proteins from string and try to target them instead. To conclude this unit's practice sessions, I would like to review a computational approach for drug search that we saw here. We start from a pathway and focus on a gene, understand the gene's associations, and find drugs that target it. We can find relevant companion diagnostics in order to check if the drug will be effective for a patient. If there are no companion diagnostics, find other drugs for genes from the same pathway. When we understand that genes do not operate alone, but are part of pathways and interact and associate with other proteins, we can design better drugs and have better understanding of biological conditions. This concludes the systems biology practice sessions. I hope you learned new things that can be valuable for you in the future. Good luck!